140 learning objectives. Labs that take forever to teach. High-stakes testing at the end of the year. Wait, they can't just memorize a bunch of stuff and pass the test anymore? Sorry kids, you actually have to think. Teaching AP Biology is a beast.
Teaching AP Bio is so incredible. You get to see these kids grow in their ability to learn and get an excitement for these really complex things like electron transport chains and all sorts of stuff like that. Teaching AP Bio is amazing.
Welcome to From Surviving to Thriving 2.0 Beta, making strides to an amazing AP Biology career. I'm Jenny Sarna. I'm a high school science specialist at Chicago Public Schools.
I'm Beth Dixon. I'm an AP Biology teacher from Western Sierra Collegiate Academy in Rockland, California.
And I'm Valerie May, and I'm an AP Biology teacher at Woodstock Academy in Connecticut.
We met in the AP Biology Leadership Academy 3 years ago, and we decided as newbie AP Biology teachers it would be really fitting to bring together some of the amazing minds, both here in our audience and here on our panel, to talk to you guys and have a dialogue about what really good AP Biology teaching looks like.
So now that you know a little bit about us, I've been teaching AP Biology for 3 years and in my 3rd year, and I know where I feel like I am on the AP Biology Rockstar continuum. It's about a 3, because I'm a groupie. I steal from the greatest AP Biology teachers ever. Good job with that. So with 1 being, oh my gosh, there's a framework, and 10 being an AP Biology Rockstar, take a second and rate yourself from 1 to 10. Where do you feel like you fall? When you have your ranking, put your ranking up so that we can see it. And panelists, you are welcome to join in.
Ready? Negative.
Some of them are underestimating.
I know, they always do that. Thank you. So we're going to run through a few survival tips from our novice experience. There are so many great opportunities out there to stop feeling alone and make friends. You're here in this room surrounded by AP Biology teachers. It might not be uncommon that some of you have more AP Biology teachers in this room right now than you have in your school district. Raise your hand if that's true for you. I mean, your state.
My country.
The amazing thing about teaching AP Biology is it is a community. From the online AP Biology, aptly named community, to opportunities like the NABT conference, which brings together that really special group of biology teachers who teach AP Biology. There's also the NABT ecosystem, another online community. And so one of the most important things is to make sure you don't do this alone.
The second thing on our top 10 countdown, you can tell Jenny and I modified this since it started. We went from 10 to 3, and that goes with the second tip that we have as newbie biology teachers, which is allow your labs to evolve over time. In the past 3 years, I decided to pick one big idea as biotech and start my focus that way into building that into my curriculum. Year 1, I sought out a place that would teach me how to do the biotech stuff that I didn't know, so that I could figure out what that would look like for my classroom. I found a place in my community that does outreach for biotech, and I was able to go to their workshop and learn about it. It was at the very end of the year, and it was an end-of-the-year unit. In year 2, I worked with the same partnership to bring them into my class, and I integrated it into my unit within the framework, so it actually made sense in the order I was teaching it in, instead of tacked on at the very end. And in my 3rd year, this year, I actually reached out to UC Davis and said, hey, we have this great Winogradzki column thing that we're doing, and we're looking at DNA. We've had experience doing some great barcoding activities with a corporation down south. Would you be willing to work with us? And they reached out back and said, yes, we would. So we're building from what started as, I have no idea how to use a micropipette, to now we're partnering with Davis and working on building something that's doing real science in our classroom and helping students to create mentorships beyond my classroom. So it's okay to start on one thing. and then just grow a little bit.
Our final survival tip, and this is probably obvious, is to get inspired. It's not an easy job. You're probably going to be at the grocery store or in other places, you know, that feeling at 10 o'clock getting the last minute things you need for your lab, or at school an hour and a half before your colleagues setting up the lab. And so the reality is you need to have a place to draw that inspiration from. And that's sort of the reason we put together this panel today. So without further ado, I'd like to introduce our panelists to you. We brought together over 100 years of AP Biology.
I think I'm most of them.
She is
No doing of math, but they are beautiful.
Oh, I'm doing math. We got to do math now in AP Bio.
They're beautiful and happy, and here they are before you. Panelists, would you introduce yourselves?
Yeah, I'll start alphabetically. I'm Paul Andersen. I'm teaching the mean of 100. I've taught for 20 years. I started teaching in what?
I'm just laughing at you calculating the
I started at a rural school in Montana, so I started in a school where we had 58 kids in the whole high school, and I taught all the sciences, so from life, physical, earth, biology, chemistry, physics. I also drove the school bus, and I coached multiple sports, and I would direct a pep band when we would have tournaments, and so I did everything there for 7 years, and now I've been at Bozeman High for the last 13 years. I also put videos on the internet, and that's it.
And this year
This year I'm taking a sabbatical from teaching, and it's been wonderful. I'm sorry, but in the first, I know, so I don't know if I should still be on the panel, but for the first few months I would always say I miss the kids, it's really hard, but there's a lot of things about teaching that I don't miss, and so it's been a really fun time.
and the teaching of teachers. You know,
And the
you're
teaching
talking
of
about
teachers is it's wild, yeah. You've done some of this, and I didn't really get it, is that you can work with students, but they're teachers, and they get your jokes a lot of the time. And also, I think you can touch a number of different kids. And so, just to be totally honest, I don't know what my future holds for me, but I know I'm going to be a teacher.
Alphabetically, I am in Brokaw. I teach about 10 miles from here, so welcome to Lake Effect Snow. I teach in a small suburb just west of here, Rocky River. I've been at Rocky River High School for 22 years. It was my first job out of college. And I knew a good thing when I had it. It's a wonderful school district, so I've stayed there. I do some curriculum development and professional development through the Howard Hughes Medical Institute's Biointeractive website. I am in the AP Leadership Academy Cohort 1.
And I'm Cindy Gay, I'm Paul's mom.
Really awesome.
I live in Steamboat Springs, Colorado. Teach at Steamboat Springs High School right now, AP Biology, General Biology, and Anatomy Physiology. This is my 33rd year teaching. I started out at a super high-end private school. Left private school, taught at Seawolder for a while. Opened a high school in Cherry Creek School District, east of Denver, and then moved to the mountains, taught at a 2-year community college for a couple of years, and then ended up at Steamboat Springs High School. So it was a very random path.
I'm Valerie May from Woodstock Academy in Woodstock, Connecticut. I've been teaching for 19 years, which I'm still 28 because the kids in front of me are always the same age. Right? And AP Bio for the last 7 years. And I'm part of the BSCS Teacher Academy Cohort 1.
All right, I am Kim Parfitt. I am from Cheyenne, Wyoming, teach at Central High School. I come from a non-traditional background for teaching. I have only been teaching about 12 years and 6 or 7 years with AP Bio. Prior to being a teacher, I worked for the Nature Conservancy doing conservation, and so have a background in natural resource management. I have taught everything from astronomy to AP Bio, and botany and zoology in there as well. So my colleagues used to joke that I would have whiplash because I was going back and forth between looking up, looking down, looking up, looking down. Telescope, microscope, telescope, microscope.
Thank you. So here's how this is going to work. We have 6 questions prepared. We're going to allow our rock stars 7 minutes per question, so they're going to decide who's going to answer what. And then it's going to leave plenty of time at the end for you guys to ask your own questions. So as you're listening to, feel free to jot down ideas to anything that isn't asked in our questions that ask at the end. Aren't they beautiful? Okay, question number 1. There's success and failure. What failure in your AP Biology course have you learned the most from?
You want me to go?
Any of you can jump in at any time. You have 7 minutes total to discuss this question. We'll listen to all of you. You can think about it.
I'll start. The biggest failure was the first day of AP Biology, quite honestly. I underestimated how much work it was going to be. I had taught all the sciences for 7 years. I got to come to a new school, and they asked me to teach AP Biology, and I met with the other AP Biology teacher. It was the day before I started to teach, and we were planning where we were going to go, and he was saying all this stuff about Hardy Weinberg, and I had no idea what he was talking about. And that night, I really felt like, what have I done? I'm really over my head. And that night, it was a continuing process of 3 or 4 hours every night, just going through Campbell and making sure I could get it and then present it. And when I look back on that and I think about those kids I had, I just feel so bad. Yeah. And they'll say, I love your class, it was so great, and you have no idea how bad I was and how I was just trying to be a little bit ahead of you. And I just found it to be incredibly humbling. That's my biggest failure.
So if I can piggyback on that, yesterday a woman walked up to me and she said, "Hello, Miss Brokaw." And I was like, shit. I wasn't going to use profanity. And I recognized her as a former student, and she said, I'm in my 14th year teaching biology. I had you in 1994-95, and that was my 3rd year teaching, my 2nd year teaching AP. The end of my 1st year teaching, which I was not teaching AP, my school district approached me and said, we'd really like to start AP Bio, we'd like you to do it. And I didn't have another AP Bio teacher in my building to even meet with the day before. And I totally just related to everything that Paul said. You're doing just enough to stay ahead of the kids. Ashley, my former student, I just looked at her yesterday and I said, oh, I wish I could have you back in my classroom now. Please come back to high school for just a few days. Because it's such a lack of confidence. It's surviving almost an hour at a time, not even a day at a time. And I'm not saying that is a failure, but that was a pretty difficult time in terms of the confidence and the material and surviving, keeping your head above water. Sometimes it wasn't above water, but.
So I've had a lot of failures in 33 years, but I really believe it's sort of like gardening. If you walk up to the garden, you see what's growing and thriving. You don't see the 100 of plants that have died and decomposed under the soil, making the soil richer for those plants that are growing, right? And so, I can't help it just oozes out. But I think for me personally, my biggest failure on a long-term continuum has been the failure of time. And so, it's April and I still have human body and cell respiration and cell signaling and still to cover in this 2-week window, right? Study hard, learn fast. And so from that I learned, you've got to plan and budget. But it was, for me, the failures, okay, so I'm not good with just one failure. So I had this failure of not enough time, right? So then the pendulum swung clear back the other way, and that I have a year-long calendar. So I know where I need to get, so that it's not May with, 5 weeks of stuff left to teach in a week and a half. But I went the other way. And I went so far the other way that I had, okay, if I'm not to this by Friday, I'm behind schedule. And then I lost the spontaneity. And so now my failure is sort of waffling somewhere in the center range in that, because what I did when I went this way, I lost the beauty of the interconnectedness of biology, which is part of its allure in the first place, because I was so tied then to getting done. And so I gave that totally up, and I just kept a checklist. And so yesterday a kid came in and she's like, I just read Henrietta Lacks, and somebody else had read it, and somebody else hadn't read it. And so there went half my hour-and-a-half lesson with kids talking about the concepts in the book and the ethics. And so now I just have a checklist of learning objectives. And so I'm like, okay, we have just talked about, my friends, this learning objective. So when we get there, I'm just going to remind you that we had this beautiful discussion today. But I'm getting more comfortable with the shifting of time and still trying to get things done.
So do you have one more?
For me, the biggest failure, again, it's kind of hard to say as a failure, but I made some major assumptions about who my students were and how they learn. I made the assumption that that they would be highly motivated, that they would be that they knew how to study, and that, I kept Campbell and Reese on my nightstand next to my bed. Of course, they would too. And that assumption was quickly slammed down when I was doing some kind of meta moment reflection, and this boy wrote, "I don't like assigned reading." And at first I was just like, and then I realized none of them do. How do I get them to do this reading? I really thought that if I just said, read these chapters or do this work, that they would do it. And I think once I realized that they are just learners like every other learner, that all that good pedagogy needs to happen again. And once I started to go where they were, it made a huge difference.
For a second question is, what's the most useful advice another science teacher has ever given you?
I'm just gonna. Do you guys have meetings in the morning when the parents come in and you have to meet with the kid? You know what I'm talking about? But you have to go teach in a half an hour. And I don't want the panel to be that, where we all sit down and then each teacher gets to talk. You know what I'm saying? Because you're always just waiting for my turn to talk. And so go free for it. Go for it. Whoever wants to jump in next, don't feel like we have to. Because Ann and I will talk all the time. And so I think, so whoever thinks they have a good answer to that should go.
I had one. Go.
Early on, I was being really, this was before AP Bio, just early on in teaching, being really hard and critical on myself and taking everything personally that was happening. And a teacher told me, a good friend said, be good to yourself. Just be nice to yourself, because if you're not nice to yourself, then there's probably nobody else going to be nice to yourself. And just know that you're going to get the kids where they need to be, and you're going to make mistakes. And you can fix them. Nothing's permanent. You're not damaging them for life if you teach them the electron transport chain wrong and then realize, like, wait, I got to go back and fix that. And that just to think about teaching as a growth process, that you're going to get better, and you don't always have to be comparing yourself to everybody else, as long as you're working hard on what you need to work hard on. And then also have a life outside of what you're doing, and get some exercise, and go for walks, and be with your family, and do things like that.
So my best advice actually came from my art teacher. Can I say it? Mm-hmm. It's not the science teacher. She is an amazing art teacher. She has ceramics, so she's got a lot of prep. She's got a lot of stuff to do. But she said that she told me to not work harder than my students. And I know that sounds like a really unrealistic goal, but it's so true that when I let the learning happen in my classroom and let them grapple and let them work, it pays off in spades. I still have to be deliberate and thoughtful and do a lot of front loading, but they're the ones, they should be working just as hard as I am, and they need to be working. And so that was really important information for me.
So I have to tell you, my daughter is student teaching, and somebody gave her some advice, and it was actually the same advice that I most valued. And so the advice that she got was, you have no idea. And the advice that I got was, we are all, us and our students, in the process of becoming. And so the you have no idea is, it's my mind fills with Thomas. Thomas was in AP Bio and getting behind and looking sleepy in class and, just sort of like putting his head down, like really disengaged. So taking it personally, all of these things. Turns out that Thomas's mom had lost his job. He went from high school to work and worked until 2 a.m., got up at 5 a.m., and ran a paper route to make a little extra money to help keep his family in his house. We had no idea. You have no idea if the teacher across the hall had a fight with whoever in the morning, their kids, their husband, their whatever, or this just happened, or they got the horrible phone call. You just have no idea. And the kids are all in the process of becoming, and it's not about the 5, as much as we want it to be about the 5. It's about kids becoming adults. It's about kids becoming ready for college. It's about kids becoming biologists. And they're going to do that at their own rate and their own pace. And so we have no idea. But it's about nurturing and helping them and us become.
The best advice I ever got from another teacher was from my father. He was a math educator for years and taught. He was a college professor as well. And I got the chance to take his class when I was in university, which was really fun. Because I got to see him, and it's, you know how you take on this different persona when you're in the front of the classroom? And it was like, I'd never seen this man. And I remember him just saying, you have to ask kids questions. Like, you have to figure out where they are. You're never going to get where you want to go unless you know where you are. And that's something that I go back to the more and more I teach, is that when I was early in my career, I just was talking all the time and explaining all the time. And now I find that I learned the greatest lessons in silence, where I could just be quiet and have them explain it to me.
Mine's quick.
You have plenty of time.
I know we're being timed. Mine's very, very quick and very, very simple, and somebody said it to me my first year of teaching, and like I said, I've been in the same building the whole time, and I still today live by it. It was a math teacher, however, not a science teacher. He said, Annie, do the best you... I'm getting a big lump in my throat right now. I'm totally a sap if you don't know that about me. Do the best you can today. Teach the best course you can this year. And strive to be better next year. And I still do that every day and every year. I want to be better the next year.
All right, let's get to the practical stuff. Enough of this feelings. What is your favorite lesson or tool for teaching a certain topic?
I can go. Because I love them so much. I love it when kids do the butt cheek shift, right? Where they have a change in their thinking and they go, what? I love that. I love that. And so anytime I can do something where, or they go Sharpay, right? Those little dogs that are all wrinkled and they go. So I look for lessons that do that provoke that. And the one that I think I just love doing every year because I can just know how it's going to turn out, and that's working with the amino acid starter kit from MSOE CBM, the Center for Biomolecular Modeling. It takes really complex ideas and turns it into a foam tuber and little clips, and the kids, they're working through it, and the little light bulbs go off, and they're talking, and there's discourse, and all of a sudden they're done with it and they totally get it. And they're so excited and they feel so efficacious and they can't wait to show off their knowledge. I love it. I look forward to it.
You repeat that.
What was the name of it?
It was the amino acid starter kit. Oh, the butt cheek shift. Yeah.
No, that's not what
So they'll add 3D molecular design. They're down in the exhibit hall, and they've got all sorts of stuff for pretty much everything. And I'll just kind of go off that one. Using models and animations and things that kids can see. I just remember, I was an extremely visual learner. I would sit on a test in college and know that there was an elephant on the page, but can't remember what the actual content was. So I saw stuff. And I now see these animations from all sorts of places and these models, and I'm starting to get it better, even after teaching it for so long. And what really told me that was so important was these kids more and more was like, oh, now I get it. It's like, I've just been talking about that for an hour, and now you get it? But it's caused me to kind of step back of that talking and more of the seeing and the doing, and which helps to put things into their place.
Do you have a specific animation that really stands out that the users see?
Well, there's the DNA replication one that's showing the leading and the lagging strand. And I remember watching that and oh my God, now I finally understand this Okazaki fragment stuff. And the leading and the lagging, and I was so excited. And I showed it to the kids, and we had struggled through it for 2 days before that, or a day, and they were excited. And now, though, the kids, they don't quite see that excitement because they haven't done the struggle. So I think it's still important to have them struggle through something first, then pull out that visual, so they get that aha moment instead of just another moving picture.
So what do you start with, Val, when you do that? When you give them the struggle and then give them the second part for your Okazaki fragment struggle? When you're giving it to the kids?
I use poems a lot, and which they're independent and they're kind of working through it on their own, or write an explanation, or give them a question to think about.
Okay. Thank you. I had to know.
I could jump in for a sec. Sorry.
No,
I'm a huge believer in inquiry, and I've always been a huge believer in getting kids to do and design their experiments and So for me, I like to start with that, and I use an enzyme lab. I just use a yeast and hydrogen peroxide lab, and the reason I like to do it at the beginning is it's one that you can do and get inconclusive data if you're not really precise in the way that you design the experiment. And I want kids to know that they're going to bring me their results, and I'm going to say, this is not working. You got to do it again. And there's always that look when you say, we got to do this again. They're like, they've always done labs where they just follow it and then you're done. And the idea that it could continue or, yeah, so lots of times it'll come down to they didn't wash off the forceps correctly and that screwed it all up. But if you've done research, that's what science is like. It's not following a path, it's troubleshooting where your errors are. And that's a message I want them to get right away.
I would say, I think rather than, okay, so I have my favorite bags of tools, the 3D physical models, kids holding something, turning it, sighting down an aquaporin.
Oh, yeah.
You know, there's something about touching stuff that matters. HHMI, the videos, the animations, the click and learns, but also some of the other pieces, some of the other things that I just have to put in a little plug, some of the pedagogical structures and tools from BSCS and the AP Biology Leadership Academy, the explanation tool changed my life. I just have to. And so for me, they're all tools, but giving kids opportunities to have academic discourse and modeling that and asking questions themselves, and I'm asking questions and we're sort of this community of learners. Learning how to learn is probably the most important tool I can share with my kids. So, as a question, multiple choice question of the day, they got to clicker it. The answers of the 5 choices came out 25, 25, 25, 25, 0. 10 minutes. Dig into your books, talk in your small groups of 4. You have 10 minutes to figure out what the right answer is. Next clicker, 33, 33, 33. Like clearly we still don't get, but the conversation was so rich. And I'm like, okay, we're gonna be, it was about endosymbiotic theory or something. And so we went out in the pod, or the little hallway area, and we modeled it. We stood in a circle and we were cells, and then there was a bacteria and they had to get engulfed by the other people and the insides became the outsides and vice versa. And then we went back and we just sat down on the floor and talked about it again. But it was their conversation and them grappling with it that I think those are the most powerful tools for me. Another tool?
I was going to say acting. Yeah, I was going to say acting things out. It wasn't. It's not necessarily my favorite tool, but that, as you guys were talking, that's what I was thinking. But then you said that about endocytosis. I think is what you were talking about. But yeah, I have to. It's about conversations. It's my classroom is much more now about conversations. And like on Biointeractive, there's a really great gene regulation modeling activity, and it's the Stiglback PIDX1 regulatory story. But I love that activity because the kids they go into these groups, 3 or 4 kids. And all I do is move. It's kind of funny. About 2 months into the year, the kids know that when we break up into groups, they always have to just have an extra chair at their table. Because they know I'm just going to rotate from group to group to group. So they have like Miss Brokaw's chair. And I love just, during that activity particularly, I just go from group to group to group. And I know it's very hard for you to believe, but I rarely say anything. And I just listen to their conversations and how they teach each other. Or somebody will say something and then somebody will say, well, I'm not really sure that that's how it works. Or maybe I'll say one thing just to redirect the conversation, but it's more about conversations with each other and learning from each other.
Okay, our next question is, which lab was most challenging for you to learn as a new teacher or now? What was the most challenging lab that you remember?
I'm going to start. Every single one. Because every single time, the first time I feel like I do a lab, there's some like major failure that takes place. It's supposed to turn yellow as the reaction goes on, and they all say, "Mrs. May, they're all yellow right now." We haven't even started anything. So learning to read the directions, I guess, is important. When there's a certain order that you're supposed to make up the reagents, and make them up in that order, so you're not mixing your enzyme with your substrate. But so it just, each one was a struggle. And going to workshops where you did a lab with someone to kind of walk you through it was great. And then there's always those ones where you're just having to figure it out on your own. But then they get better and better every year. Because you know the tricks and the trade, tricks of the trade.
When I first started teaching AP, it was all the biotech stuff. I hadn't run gels in my undergrad, and I hadn't done my graduate work yet. So there was a learning curve for me for just some of the content and lab equipment. I had very little experience with micropipetters. And you know, some kids put a tip on a micropipetter and they're shaking so much that they can't even put the tip in the microcentrifuge tube. And then you're trying to show them and you're shaking too because you have really little experience. So I would say the biotech stuff for me was my early on, that was steepest learning curve. With the new labs, it's just some of the new labs, just learning them and forming a network of people, like on the online community. If you're not on the online community, join. Just sucking ideas from everybody else. You know, post, I'm having trouble with this lab, can anybody help?
Yeah.
Okay.
Do you want to go ahead?
Well, we were talking this morning about this question, and I agree with Val that they all can be difficult, and especially the ones that have a lot of equipment that is, sometimes it gets in the way of the conceptual understanding that you want the kids to get. But so there are so many resources in terms of people, and also with HHMI and all these other resources that people have, that sometimes I feel like the lab, that I can scaffold it with these other experiences, or that there are other, an accumulation of other experiences that can get me to the same outcome, the learning target that I need the students to have. But the only way you learn to do that is to go for it and see how it's going to work out. Sometimes I'm always really surprised. I'll read through the lab and I'll go, oh, this is stupid. And then I'll do it with the kids, and the kids are like, oh, look. And I'm like, oh, I guess I'm the one that's stupid. but so I guess, you know, be flexible in your thinking and be open-minded. Go for it and learn from your mistakes. And just like Ann said, every day is a new day, and I can get better the next day, and I can get better the next year.
And if I could just add one thing, and if you're doing the lab for the first time, tell your kids that.
Absolutely.
So I love my kids to know that I've never done this with a whole group of kids. We're in this together.
Absolutely.
Well, so that's my funny story. Once upon a time, there was a teacher who never went to teacher school because she got hired right out of undergrad to teach at a private school, and you didn't have a teaching license, right? So that teacher she was still living with her mom and dad because she didn't have any money, and I took home every stinking lab, and I did it at home on the kitchen table. Actually,
Mom and Dad,
with
that's
Mom
my
and Dad, they
students.
were the students. I know, like, so my family
I love
still.
this story.
I do. So at one point, I don't remember even which lab it was, but you needed dark, like, I don't even know if it was a transformation something. My whole family was huddled in the bathroom with the experiment on the tank of the toilet to try it because I felt like I had to have a lab, I hadn't had it nailed, and you know, then finally I'm like, this is really stupid. I could do this with kids. So I used to, before, like labs that I'd never done or I didn't feel very comfortable at, I would run my trial alongside the kids and we could compare. And now, sometimes, if it's a lab, the very first time I ever grew a fast plant, I was like, well, this is stupid. I'm not going to do it first because it's 36 days, right? I don't have 36 days. They grew them, and they gave me feedback on the lab protocol. So I became the student, and it was their job to teach me about what worked for them, and that was actually sort of cool.
And asking kids to do that, like give feedback, that's them thinking about the practice of science.
Yeah. Yeah, and making mistakes and like, well, what if you did this? And I'm like, I don't know, let's do it.
Let's try it.
Yeah. I'd find myself in the middle of a lab, and Kim, you said something that resonated with me. Do they even know what's going on? I was doing the photosynthesis lab, and we had DPIP, and I organized all these spectrophotometers, and I'm in the weeds, you're in the weeds, and what's the point of doing, what is the point of doing this? Is it just so you can run a spectrophotometer? I would argue that it's about the practices of science, and so for me it was trying to get those labs to a place where I'm happy. Like I started using the leaf lab instead because they could physically see a leaf that I cut, and now it's going to float. And so that's what I've tried to do is, I don't want to be in the weeds again.
Right. And that's what I think you have to be real deliberate about is, and this comes, I think there's a lot of pressure when you're starting to teach AP to teach it right.
Right.
And, to try to, I used to think that I was supposed to get through that 40-pound book, and I was just panicking. And there isn't that right way. And so you put a lot of pressure on yourself, and you need to step back and say, what is going to move my students best? And make choices. And your students will appreciate that.
So, 40 to 50% of teachers leave the classroom within their first 5 years. What kept you guys going for all these years? 100 plus.
I just got grayer from that.
And when you look at when you were in that spot in those first 5 years where it's hard, what was the moment where you decided, I'm going to push through and continue with this? What got you through personally?
I'm going to jump in again first. So I think that in that first 4 or 5 years, you're really not seeing the outcome of what you're doing. The kids, especially if you have sophomores, are still in high school, and they don't quite know yet what value they got out of your class and out of the experience. But when you get to that 5th, 6th year, you now have kids that are coming back at Thanksgiving and saying, I have the highest grade, or I'm doing amazing in my biology class, while everybody around me is struggling, and it's because we learned this with you. So in those first couple of years, you're not getting that gratification, you're getting a lot of the struggle. And if you just kind of wait through that, and have the kids start coming back, and not all of them are going to come back. It only takes one kid to come back and say thank you. And then, it's like what the politicians say, for every one letter that represents, 10,000 people thinking the same thing. So every one student coming back, it's 10,000 students saying the same thing.
I'd jump in just for a sec in that for me it's my family, it's my wife, and it's my kids. Like they allowed me to be crazy at night and just study and study. Mark Little is in the back, and last year you asked me to come to NABT, and honestly, I'd never heard of NABT. And I just learned what HHMI was. So we're from Montana, and there's not, we don't hear about a lot of these things. And now, to kind of come full circle, I think about that, and I always worry if you're a new teacher to come to something like this and see exemplar teachers, that I don't know if that would have dissuaded me from the hard work it takes to get good at teaching. I think that 10 years of just toiling away, learning the content, learning how to teach, that's where you should, don't change the world, don't change the education world, just become a better teacher. And like what Ann said, it resonates with me. Like, is it working? If it's not working right now, change it. Not tomorrow, but period 2. Like, never be willing to try new things, but if it doesn't work, iterate on that.
You know, I think for me, it was honestly, it's the story of biology. Just every year, every summer, I would cram in as much professional development as I could find. And it was the good, juicy professional development. You know, it was a combination of content and pedagogy. And the deeper I learned, the more I wanted. And so I love the stories. I became a voracious reader. And when I'm able to tell those stories in class, you know, I get all excited, and the kids get excited because they see my passion. And the stories just get better and better, and then when you can back those stories up with incredible animations and using the grants data from the finches on the Galapagos, I mean, it just is so rich and beautiful. It's this great creative endeavor. And then you throw kids into the mix. Holy smokes, it just doesn't get any better.
So it was my 5th year of teaching. I was tired. I was really tired. And I remember driving in the car, I don't even know where I was going, I know I was driving the car, and I realized that, okay, I'd spent 3 years in middle school, 4 years in high school, 4 years in college, I'd spent 5 years teaching. Like, that's the longest time I'd ever done anything in my whole life. Right? So I'm driving my car going, I gotta do something else. This is like, I'm pooped. And I'm thinking, okay, great. What do I want to do? I love biology, I like kids, I could do this, I could do that, I could do that, I could like have a job where I could help people really understand how cool the world is. And I'm like, and all of a sudden, I know this sounds really stupid, I was like, oh my God, that's what I'm doing. I know it. But really honestly, I still have those moments. Like, I know, right? Some of us are slow learners. But honestly, you know what I would say? I would say, put a seed inside a paper towel in a glass and watch that seed come to life. Grow a garden. Take a walk. I don't know if you're like really into molecular stuff. I don't know, do a gel. But, I mean, like, really, what brought you to the profession in the first place? It wasn't, for me, it wasn't that I was in love with the electron transport chain. I loved nature, and I loved life, and the interconnection of life, and this, it's like Kim said, this wonderful, incredible story that you can tell from the front to the back, to the inside out. It's that love of biology and wanting to share that story. And it's the passion for the science, and it's so easy to get lost in the, oh my God, this lab didn't work, I still have to pour plates, I still have to blah, 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 and then go home and grade the papers. But that's not, it's like sharing stories, it's being outside, it's your love of life. And little kids growing like seeds, to be sappy.
Do we have time?
Yeah.
Oh,
I'm so
you know what? I taught for I have it's kind of a boring answer, but I taught for 5 years, and then I left the classroom and went to graduate school for a year. And about Christmas break of that year off, I was miserable because I wasn't in my classroom. And I knew my classroom was home. There's my end.
All right. Our last question from the panel, before we open up to you, is what advice can you give to early career teachers to help them transition from surviving to innovating?
So my dad is an amazing influence. I have the big lump in my throat again. And from I have no idea, when we started getting this message, I was probably 2. Life is about networking, Annie. Life is about networking. For that answer, to me, is if you're sitting by someone you don't know right now, introduce yourself. If you haven't gotten names and emails and contact information from people at this meeting, get them before you leave. If you haven't joined the online community, join the online community. Surround yourself, good teachers steal from other teachers. And that's how I really survived. And if somebody says, please email me if you have any questions, really do that. It's about forming a network. It's about supporting each other. I know that I can any day call Cindy Gay and say, Cindy, I had the crappiest day today. And she will talk me off of the ledge. You need that support. You participate in professional development. Even if it's within your local district, just in your department, or being on the online community, or going to meetings, or going to a summer workshop, doing some of the footwork to find some of those things. But build that network, and it's so isolating to be in our classrooms sometimes, and we have to get outside of those 4 walls.
4 years ago, I was actually at this conference, sitting in a session of Anne Brokaw's for HHMI. And after the session, I emailed her shortly after I found out I was going to be teaching AP Biology. And I emailed her and said, oh my gosh, creepy, I was in your session at NABT, and I know you're a really good AP Biology teacher. Do you have any advice? And I'll never forget, because this is probably right before Christmas break, insanely busy time. I woke up the next morning to this 3-page email in my inbox, hey, that's great, you're teaching AP Biology, and all this stuff. And I'm the horrible person. I was like, oh my gosh, I'm going to need Christmas break to read this. I get an email the next day. Did you get my email?
Are you still alive?
And I didn't even know where to start. Things didn't seem fitting. But I asked her last night, I said, I am still so touched, thinking, and now understanding how busy you must have been. I'm so touched that you sat down and you must have, the stock email, that you changed the name. And she's like, no, that's a good idea.
She said that to me last night. She's like, you must have, this, a form letter email. And I was like, no, but that's a really good idea.
And I think that for early career teachers, there's, some of that is, if you're teaching for this long, it's because, you're Cindy Gay and you've slammed on your brakes with that revelation. Or there's a real passion there and there's a real love. And so, reaching out to people.
Yes.
And, but don't forget to write back and say thank you. Yeah.
Oh, that's not why I was asking. I really wanted to make sure that you hadn't quit your job.
I would jump in. For me, the best professional development I ever did was just putting myself out there. And I know you don't want to make videos, obviously, but you guys presenting today, or what all of you do, I find, share what you're passionate about with other people, and the feedback you'll get will be honest, and it's just like teaching. You're going to learn more from doing that than the people sitting in the audience.
Right.
And if you're not comfortable yet with that, I know, when I was starting, the thought of giving a presentation would have, given me palpitations or something. It's super easy to ask for advice. Here were 6 awesome questions. We could have gone to Starbucks, and they could have just said, turn to a neighbor and answer this question. It doesn't matter. I mean, okay, so anybody didn't fail in their first year? Right? We could all talk about our failures. We could all talk about our favorite something or other, and like surround yourself with people. Everyone has great experiences, regardless. They don't have to be old, but everybody has awesome, great experiences like me thank you, to share. And so, how do you tap into that? And
So when I started teaching, because I am like that other person who didn't go to teacher's school and get all that stuff, I felt like a faker, and I felt like I didn't belong teaching this stuff because I thought everybody else had it going on. And the more professional development I did and sought out, the more I realized exactly what Paul's saying. It's you just put yourself out there, you just go for it, you learn from it, and you build relationships with people, and people help you. And I just think that seeking out opportunities, summer workshops, I know many of you might have, you have families, but even webinars or online things or reading, or in our district we do lab classrooms where people come in and watch people teach, and it's just great dialogue of what's going on. And, I always want feedback, so I'm the first one to raise my hand when it's like, who's going to host the lab? I'm like, I'm there, please, come see what I'm doing. Tell me what's going on in my classroom. And it built my confidence with my content, but it also let me see that, everybody's got it going on. Everybody's bringing cool stuff to the table.
And I would say surround yourself with positive people. I mean, we've often gone to workshops or something, and there's that person that's super, super negative about it all.
We don't have time.
Just, we don't know what's in their background, we don't know why they're negative, but just kind of move to the positive and don't get bogged down by, I remember first AP Bio thing I went to, I just felt intimidated. And I felt like there was people who were intentionally trying to intimidate me.
Yeah.
And it was really negative. But over the years, I've surrounded myself with some super amazing people, both out there and here. So it's great.
Before we open this up to questions from the audience, thank you so much. And I know that this idea to host this conversation was because of conversations that I've had with Val and Cindy and Ann during our Leadership Academy and through BSPS and NABT especially, because before NABT, I didn't know there were teachers like Val and Cindy and Ann. I thought that they were, really far away, like Paul Andersen.
So after our question session, we will be selling Polaroids for $5. You can come stand behind the camera. Let's hear from some of you.
Yes, sir. Oh, I'm sorry. The facility. Yes.
All right. First of all, thank you so much for sharing your collective wisdom. And I really appreciate your enthusiasm and your passion. And I can feel it. It's inspiring. But based on how we are often measured, how our success is measured, which is the AP exam scores, I want to find out how you were able to deal with, and maybe you didn't experience this. I teach in the poorest district in Rhode Island. My students come to my class completely underprepared, have never been challenged in any other way. And so when they come to my class, I still end up with a mean score of less than 2, even though I work harder than any teacher I know in my school. How do you deal with that? It's frustrating 3 years in a row. I work and these guys say, we're doing it. And then last year, out of my own mistake, I taught them how to write an essay. They finished 2 out of 6. Because they said, oh, no, we did the outline like you told us, and they ran out of time. Mm-hmm. So how do you deal with that aspect of it? Not the passion, I can feel that.
Right.
But how do you deal with the scoring, that logistical aspect?
I can jump in for just a sec. And I taught for 13 years, and that first year, I was just waiting all summer for the scores to come back. And I was bugging the secretary because I really wanted to see how well they did. And I've always just driven myself on, I want to get the scores better and better and better. And what's happened over those 13 years is I cared less and less and less about those scores because the relevance is not there. I know how well the kids did. But I was able to be okay because I was the only one who really cared about those scores. I know there's a lot of districts where other people care about those scores, and I just wouldn't be able to teach in a district like that.
I don't have any internal district pressure for scores, and I have evolved, as Paul has. I gave a session yesterday, and some of you were there, and the thing I said was, I just really want to teach a really kick-ass biology class. It's not about the test for me. I tell my kids on the first day of school, you just walked into my classroom on August, whatever. My goal is that you are a better student when you walk out of my classroom. And if that means I'm taking a kid from here to here, or you're taking a kid who maybe is coming in with a little less background and maybe not stretching them as far as perhaps I might be stretching my higher level, you know what I mean. I want them to be a better student when they leave me. I want them to have some skills that they didn't have before. To me, it doesn't matter if they get a 2 or a 1 or a 5. I want to prepare them for the next level, whatever that next level is for that student.
This past year, I had a conversation with the students that day before they went off to do the test. And I said, all right, so this is a big deal test. We've worked on it for the content, the skills for a long time. But it's just a check. It's just to see where you are at this particular time. It's not an endpoint. A 1 doesn't mean you're done in bio.
Right.
A 1 just means that you need some more time. So if you're still interested in the things that we talked about, because our focus was on the learning and not on the outcome as much, keep going. Take that freshman bio class, and you're going to do awesome. And you're going to build that confidence. And I just have a really supportive school district with that as well. They want numbers, not necessarily scores. And that's really, like, if your school is really worried about calculations and ranking, the ranking isn't based on how many 5s you get. It's really on your enrollment and who's taking it. And maybe remind them that that's the importance, is who's taking it and the experience the kids are getting out of it and what they're going to take forward.
I teach at a school where the first day of school, the principal puts our scores up in front of all the faculty. I teach at a school where the AP's test scores are in the paper. And obviously we know who the teachers are because there's only one AP Bio teacher. So accountability is huge right now, and it's very uncomfortable. I was fortunate that my principal basically said, for the first 3 years, I'm not going to look at your scores, even though we still flashed them up in front of the whole faculty. They are a point in time. I think other measurements, for example, we are also held accountable for ACT scores. And so I make sure that I'm also monitoring that. And so I have another metric by which I can show growth. And then we keep those little letters from students, the email I get from, Logan, who's saying, I have to tell everybody how to do chi-square because nobody knows how to do it at the University of Wyoming in the biology freshman class. And he knows how to do it. So, maybe finding, because obviously you're at a school that cares about those scores, so maybe find some other metrics that help show a broader picture.
Well, if I can just comment, it's a matter of, they thought I was crazy teaching these poor kids immigrant kids, AP Bio. They
You're
thought,
not
and so
crazy.
unfortunately, I haven't been able to disprove. I mean, I got a kid into MIT, Harvard, Brown. These are things that happened in the last 3 years, but I haven't been able to come back and say, you see, look at the scores.
Yeah.
Because even though I get the 5s and the 3 and the 4s, most of them are still scoring 2 and below. So I was wondering more specifically about, do you train them to take the test? I mean, I feel like that's my thing.
So,
Yes.
that was actually, I was going to answer,
Yeah.
believe it or not, a little more pragmatically for me than I usually do. But I think, because a lot of times my kids are like shell-shocked as well, I think there's some merit in going slow to go fast. And I, honestly, the whole first quarter, I don't cover a lot of content, but I cover a lot of learning how to learn. How do you read a textbook for understanding? Like, they don't even understand there's an internal structure of headers and subheaders, and how do you go, and you don't just start at the beginning of the textbook. And how do you learn how to learn? What do notes look like? Who are they for? A lot of those learning how to learn skills buy you so much time later because you're not continually fighting that battle all year long. And the other thing I have done with great intent is, what are those things in my course that recur over and over and over, that if they don't have that basic understanding, and it's going to be different in every setting. For me, if I invest heavily in protein structure at the beginning of the year, I mean, seriously, I spend like more time than anybody would think humanly possible and percentage-wise on protein structures. Cool. Well, A, it's cool, but B, I know that time I'm spending up front going super slow is going to buy me boatloads of time when I get to cell signaling, when I get to genetics, when I get to, evolution down the road, because it all somehow weaves back to that. And so picking those few things that if they just knew this, all these other things would start to make sense. And so that's a pragmatic thing. I always start the year with a throwaway lab. My very first real lab is the enzyme lab. And so I always start with the Bradford assay. And I tell them, this is our throwaway lab. We're going to get crappy data. Oh no, we're going to do it right this time. We're going to be the year. No, they always, but they're learning from that failure. The other thing that I have done is starting to have conversations. Well, okay, I am the general biology teacher too, but having conversations with what are the skills collectively? What sort of learning how to learn skills, lab skills, science practice skills, could we distribute to the end of whatever class comes before, or even the beginning, so that there's some continuity in these skills and you're not starting from scratch?
Could I jump in once? And I would encourage you, instead of looking for the mean, which is I think what you brought up at the beginning, you should be looking at your end value. How many kids am I getting into the program and getting them successfully through the test? And also, do I teach kids how to take the test? Of course.
Yeah.
Yes,
Like,
absolutely.
if you don't give your kids a good idea of how the test is formatted, and if you're not aligning what you teach to what the College Board would like you to teach, you are setting them up for, of course you teach to the test. Difference, this is a really good test.
Absolutely.
There's a good policy.
So just, I don't know your name. Can you make sure that you get a couple of our emails? Because we can offer some strategies too that you can use, like practical things. And Paul has a really great video online about the test.
No,
And
my students
so,
have watched it, and he's getting to my school.
but we can give you, like, we can all give you some other things.
Oh, that's a white guy. Oh, that's smart white guy.
I'm going to talk to that now forever. Smart white guy.
Next year, that's his badge.
I'll take it. There was a question over there.
Yeah, and so unfortunately we're coming up on 2:15, and I want to be respectful of time in this space as we finish at 2:15.
And
But I
then
think this.
2:30 session. Oh, yeah.
Are we have time?
We can do one more.
One more question right here.
Well, it was more a comment geared towards what you were talking about. I'm from Alabama, and we have a tremendous amount of immigrants as well as just across the board, poor students whose families have never even thought. And what it sounds like to me that you're struggling with are skills, not content. I'm sure you're an amazing teacher and you're doing a phenomenal job with that. But what you have to do, particularly with this new exam, is teach skills. And the number one skill that any kid will coming out of an AP exam, and I support close to 300 AP science teachers in physics, chemistry, biology, and environmental science. The number one thing that the kids say is, I just didn't understand what the question was asking. And you explain it, content like what goes on. And they're like, oh, I knew that. So the problem is kids, particularly low-level readers like you have, is they're struggling with reading these new, longer, and more justification type. So my suggestion would be start your day off with just one question off of one of the old released exams that's in the new format. Give the kids roughly 2 minutes, and then spend about 8 minutes debriefing that question. What was the question asking? Was there any vocabulary that was difficult? What was the correct answer? Why are the other
answer? What is this question about?
Yeah.
Right? What is this question asking? And then what do you think some choices might be? I don't even understand.
So where you're struggling is your kid's ability to read the questions. And then the other thing is I would spend the last part of your day on a writing. And the way you develop writing for these new short answer is you've got to put a limit on the number of sentences that the kids can write. So that way you force them to think descriptively about the words that they choose. And
I think this is a really great example, too, of all the collective knowledge that's in this room.
Right.
So we are recording this session. The AP community, people who couldn't be at this conference, were curious about sort of the conversation that would take place in this room. So I think it's, given that there are still more questions, and you know who you guys are now and you're on the community, just to look out for that, the way we're going to get this recording and continue this conversation.
And if you're sitting there still with questions and you see us out and about in the halls, stop us, ask us.
So before we go, are there any final thoughts? And thank you so much.
My final thought is hang in there if you're just starting out. Just hang in there. It gets better, I promise.